I'm Neil Singla. I'm the Chief Scientific Officer at Lotus Clinical Research, and I was invited to speak about a paper that I wrote with my co-author Timothy Rogier on the abdominoplasty pain model. There's a lot of data that's being produced on new analgesic candidates in the abdominoplasty model, and I think that clinicians are going to begin seeing that data very soon. And that's why the paper is important, to kind of describe to clinicians why was the model chosen in the first place, what does it mean, what does the data that comes out of the model mean, and how can they interpret it and fit it into their clinical practice. The first question that's addressed in the paper is why we need the abdominoplasty model in the first place. And the basic answer to that question is that in order to understand new drugs, you want to test them in both soft tissue and hard tissue. In hard tissue, we had an excellent experimental pain model in bunionectomy, but there was really no good soft tissue equivalent that gave you the same quality of experimental outputs, the same reliability, etc. And so that's why abdominoplasty was developed, and that's why it's important. We at Lotus sat down to talk about what soft tissue surgery has the characteristics that are similar to bunionectomy to allow it to be a great experimental model. Some of those characteristics are, it has to be painful enough. Patients have to want the surgery and have good clinical outcomes. Few side effects, a very safe surgery, a surgery that's easy to diagnose. And abdominoplasty has a lot of those characteristics. So after performing several pilot studies and understanding a lot of the characteristics of the model, we did our first double-blind randomized study with Trevena Pharmaceuticals in abdominoplasty. And one of the great things about that first study that we were able to do was that it had an active comparator of IV morphine, and it had a placebo arm. And so from a model developer standpoint, that comparison, placebo versus active comparator, not really versus investigational product, tells us how the model is working. And in fact, when we got the results, we were thrilled to see that the standardized effect size, or let's say the signal-to-noise ratio between placebo and morphine, was very strong, and the model could separate the agents very nicely. The next hurdle that we encountered with the development of the model was to be able to use it as a pivotal study for the registration of a new drug. We performed the study with Recro on IV meloxicam. The study was positive and eventually led to the registration of IV meloxicam. So that was a very positive next step for the model. Since that time, there's been 13 double-blind, randomized, high-quality studies in abdominoplasty, and the point of the paper was to gather all of those studies and put them into one place. So we have, first, a large master table which breaks out a lot of the important considerations and aspects of each study, pulls them into one place, and then subsequently in the paper, we move forward and analyze some of the important experimental questions that people have been asking us and we've been asking ourselves for years about the model, such as, what is the standardized effect size of the model, or what is the signal-to-noise ratio? How good of a model is it? How does it compare to bunionectomy? Does the model work better at 24 hours? Does it work better at 48 hours? What are the rescue drugs that you should use for the model? How does the model perform when you look at time to onset of new drugs? So these are all the little questions that, with all of the data in one place, we're able to analyze, take a look at, and report to our readers. At this point, the model has become very standard in the industry. There have been 2 drugs that have been approved based on the model, and there are multiple drugs that are in the development pipeline that are utilizing the model. So clinicians will see a lot more data from abdominoplasty out there, which is why we're so excited to be able to describe it in the paper. What does that data mean? What does it mean for clinicians? What does it mean for people who want to design experiments? So I hope this paper is helpful and informative to those who are either designing or interpreting clinical trials from abdominoplasty. Thank you.